there is of course, reductive amination is a very important reaction that we carry out in the lab, and it also happens inside the cell. And as you can very well imagine that we do not have sodium cyanoborohydride inside the cell to carry out these reactions, and so one must resort to other forms to do this. Let us look at the reductive amination as a possibility inside in the lab. So, what you would start out is with a ketone such as this, which then we can react this with R3 NH2, and this is going to give you an imine such as R3 H, and there is a positive charge, R2 remains intact. Now, when we have sodium cyanoborohydride, right, you can think of this hydride transfer here to give you to use the nitrogen as the positively charged nitrogen as the electron sink, and you end up with NH R3 R2 H and R1. Okay. So, this is a reaction that we can imagine very well can happen inside the lab, and now let us look at the biological equivalent of this reaction. Okay. And as you would predict, it is going to there are two phases of the imine that can undergo reduction, and it is equally likely to that either of these phases accessed, and so you would end up with a racemic mixture of amines. Okay. But nature transforms this simple reaction into a stereospecific and reversible one that is not just beautiful for its simplicity, but also for its cleverness. The reagents are actually a pair of substituted pyridines called as pyridoxamine and pyridoxal. Okay. So, we look at this reaction reagents now. So, this NH2 is over here, and this is giving you have a phosphorylated P double bond O, OH O minus, so this is pyridoxamine phosphate. This can be transformed to the corresponding aldehyde through the enzyme called as aminotransferase. Right, and it is going to give you the corresponding aldehyde, and this aldehyde is called as pyridoxal phosphate. Rest of the molecule remains the same, the pyridinium is still okay. So, this is the reaction that we are going to consider now. So, the way in which this reaction happens is that you would use a lysine residue from the enzyme aminotransferase, which is going to react with this aldehyde to produce an imine. Okay. So, this imine is actually what is the Schiff base equivalent, which can then undergo reduction to give you the corresponding amine. So, when reductive amination or its reverse is required, the pyridoxal is transferred from the lysine imine to the carbonyl group of the substrate to form a new imine of the same sort. So, the most important substrates are the amino acids and their equivalent alpha keto acids. So, here what we need to consider is that once you have the Schiff base, then or the formation of the Schiff base is going to be the key step in this process. So, once the imine is produced, now this is going to then consider it is going to react further and give you the corresponding amine. So, by using the protonated nitrogen of the pyridine as an electron sink, the alpha proton of the amino acid, which is shown here, right, then is then removed to form a new imine at the top of the molecule and an enamine in the pyridine ring. So, let us look through this reaction. So, this is the old imine that we started with, and this hydrogen is then picked up and you form a new imine over here, and since this pyridine is the electron sink, you end up with a new enamine as shown here. Okay. And now, the electrons can return through the pyridine ring and pick up a proton at the top of the molecule. The proton can be picked up where it came from, but more fruitfully it can be picked up at the carbon atom on the other side of the nitrogen. So, hydrolysis of the imine that is formed then gives you the alpha keto acid as shown here. Okay. We will not go into the details of the hydrolysis mechanism of imines. I shall assume that you guys are familiar with it. So, all natural amino acids are essentially in equilibrium with their equivalent keto acids by this mechanism, and this is catalyzed by aminotransferase. Now, reversing this reaction makes an amino acid stereospecifically out of an alpha keto acid. Okay. So, what happens is that you can imagine, or you can consider that ammonia can react with this alpha keto acid, in this case alpha keto glutaric acid, and it will form this kind of an imine, right, and this happens through several steps. Now, reduction of this imine by NADPH, as we have seen earlier, can occur, and this is going to give you glutamic acid as shown here. Okay. So, therefore, this process, which is called transamination, starts when you start with the alpha keto carboxylic acid. So, we have looked at in this process, we have looked at a way in which an alpha keto acid can be produced from an amino acid, and we have also looked at the reverse reaction where an alpha keto acid can be converted to the corresponding amino acid in a very stereospecific manner. So, amino acids frequently get used up, for example, in making proteins. So, in order for life to keep going, ammonia must be brought in from somewhere. So, the key amino acid in this link is actually glutamic acid, as we have seen below. So, therefore, a true reductive amination using NADPH and ammonia builds glutamic acid from alpha keto glutaric acid. The other amino acids can now be made from glutamic acid by transamination. We have already looked at the steps in the previous slides. At the end of their useful life, they are transaminated back to glutamic acid, which in mammals at least gives its nitrogen to urea for excretion. Okay. So, therefore, pyridoxal is a versatile reagent in biochemistry of amino acids. Pyridoxal is the reagent in other reactions of amino acids as well, and they all involve imine as the intermediate. The simplest is the racemization of amino acids by loss of a proton and its replacement on the other face of the enamine. So, here is the enamine that we are considering, and as you can think about, this is actually going to be a prochiral imine, and once the imine is going to access or the proton can react with this from the top face or the bottom face, you can consider two different isomers of the enantiomers of the amino acid being formed, right. So, if the top face undergoes protonation, you end up with this unnatural amino acid as shown here, and if the bottom face undergoes protonation followed by imine hydrolysis, you actually end up with the natural amino acid as shown here. So, how is it possible for the same reagent operating on the same substrate, that is an amino acid, to do at will one of the two quite different things, that is, one is removal of a proton and another one is decarboxylation. So, in order to understand this, we shall look at how the enzyme is positioned, right. So, the enzyme or the protein is very tightly bound in all handles, right. So, for example, this OH here is bound to a tyrosine residue as shown here, and the phosphate is bound to arginine, okay, and you have an aspartic or aspartate reacting with or interacting with the positively charged pyridinium, right. So, this holds the substrate in a very tight manner. Okay. So, then around the methyl group there are alkyl substituted amino acids which form a hydrophobic region, and even when the lysine attachment is exchanged for the substrate, all these interactions remain in place. So, therefore, the substrate is bound by similar interactions with other groups on the enzyme. Now, control over the choice of reaction arises because the different enzymes bind the substrate pyridoxal amine in different ways. So, decarboxylases are enzymes which bind such that the carbon-carbon bond that is to be broken is held orthogonal to the pyridine ring and parallel to the p orbitals in the ring. So, if decarboxylation were to occur, so this is the key bond that has to break, and this is held perpendicular or orthogonal to the olefin, right. Now, you can push arrows and it is going to result in the decarboxylation reaction, and carbon dioxide can be lost. Alternatively, if you want deprotonation to occur, then the CH is actually held orthogonal to this. So, enzymes do not speed up reactions indiscriminately. We have already looked at in detail how enzymes are able to accelerate reactions, and what they do is that they selectively accelerate some reactions at the expense of others. Okay. So, therefore, in this very simple example, what we have looked at is the positioning of the bond that needs to be broken is very important in determining the outcome of the reaction, and certain enzymes can stabilize this carbon hydrogen bond breaking or the transition state leading to that, whereas there are other enzymes which can actually catalyze the decarboxylation. So, this is a beautiful example about how by just doing a simple carbon-carbon bond rotation, we are able to access two different products. The next topic we are going to look at is nature's enols, which is lysine, enamines and coenzyme A. Okay. So, the glycolysis pathway breaks down glucose to produce energy. We have already looked at this previously, and in doing so, it produces smaller molecules for use in the citric acid cycle. In the reverse process, it allows the synthesis of 6 carbon sugars such as fructose from 3 carbon fragments. A key reaction in this step is with 3 carbon sugars to combine. So, what the process that we are looking at is basically glyceraldehyde 3 phosphate is going to produce dihydroxyacetone 3 phosphate. Okay. So, glyceraldehyde is shown here, which is basically the aldehyde or the oxidized form of glycerol, and dihydroxyacetone phosphate is shown here, which is basically the oxidation, the carbonyl is shifted to the internal carbon. Now, glyceraldehyde is present in cells as its phosphate, which is in equilibrium with dihydroxyacetone phosphate. This looks like a very complicated rearrangement, but actually it is a very simple one, where the two compounds have a common enol which then interconverts. So, if this aldehyde enolizes, it produces an enol such as this, then you can push arrows to give you the corresponding dihydroxyacetone phosphate. The reaction is effectively an aldol condensation between the enol of the keto sugar phosphate and the electrophilic aldehyde of glyceraldehyde phosphate, and the enzyme is known as aldolase. Okay. So, these two units are going to combine to give you a 6 carbon unit, right, and this product is the keto hexose fructose 1,6 diphosphate, which is shown here, which then equilibrates to give you the closed ribose form. So, no enolate is formed in this aldol, but instead a lysine residue in the enzyme helps in formation of an imine with the keto triose. Let us look at this reaction now. So, once lysine is going to interact with this carbonyl, this is going to form this kind of an imine, right, and then this can happen through proton transfer, and they allow this imine to be converted to an enamine, which then acts as the nucleophile in the aldol reaction. So, many other reactions in nature use enamines, and mostly those of lysine. However, a more common enol equivalent is based on thioesters derived from coenzyme A. So, coenzyme A is basically an adenine nucleotide on one end, and it is linked by a 5' pyrophosphate to pantothenic acid. Okay. So, pantothenic acid looks a lot, is basically like a tripeptide, and that is attached to an aminothiol. So, let us look at this molecule in detail. So, here is the tripeptide, which is pantothenic acid, here is the pyrophosphate group, and here is the nucleobase and the ribose. Okay. So, this molecule is actually abbreviated as CoA-SH or CoA thiol, and when SH, this SH is the vital thiol functional group, and all reactions we are interested in will be that of the CoA-SH. And just to put this in perspective, these are thiol esters as opposed to the normal alcohol esters that we are used to. So, this is the thiol ester and this is an ordinary ester that we are more used to. Thiol esters, interestingly, are less conjugated than ordinary esters. Okay. So, this is because the sulfur, the overlap between sulfur and the carbonyl, or the interaction between the sulfur and carbonyl, is not as strong as between oxygen and the carbonyl. And so, the ester hydrolysis occurs more rapidly with thiol esters than with ordinary esters, and because in the rate determining step there is less conjugation to destroy. Also, the thiolate is a much better leaving group. We have already looked at pKa's of various thiols, and thiols are much better acids compared to alcohols. So, another reaction that goes better with thiol esters than with ordinary esters is enolization. So, this is an equilibrium which we have looked at previously, where the keto exists as an equilibrium with the enol, right. So, the thioester similarly exists in its enol form, and if we were to draw the enol form of a thiol, it should look like this, whereas with a normal ester the enol is going to look like this. So, a thiol ester has less to lose, and it is generally more enolized. Okay. So, again the equilibrium between the normal ester and thiol ester are going to be different, and the normal ester is far less enolized compared to a thioester. So, together what we can understand is that a thioester can be hydrolyzed more rapidly, and this is attributed to how the carbonyl is more reactive, and also the thiolate being a better leaving group. Also, the thioester is more in the enol form compared to the normal ester. So, therefore, this becomes an ideal vehicle for an enolate transfer. So, the key step in the synthesis of citric acid from oxaloacetate, as we have looked at previously, is from oxaloacetate and acetyl-CoA. So, the reaction is essentially an aldol reaction between the enol of an acetate ester, which is shown here, okay, and the reaction between these two, which is oxaloacetic acid and acetyl-CoA. Now, the oxaloacetic acid has an electrophilic ketone, which is shown here, right, and this is the ketone that is going to react. So, acetyl-CoA can exist in its enol form, and we have already looked at the equilibrium is going to be favoring the enol. So, if you push arrows from here, you end up with this reaction where the enolate is going to react and give you this citryl CoA, right, and subsequently loss of the CoA can give you the corresponding citric acid. So, in nature, enolization is catalyzed by a basic carboxylate group such as aspartate and an acidic histidine. We have already looked at in detail about histidine, how histidine can act as a proton donor as well as a proton acceptor, and both are part of the enzyme, and so this makes the reaction go even faster. So, aspartate can pick up this proton here and push the equilibrium more towards the enolate, and this carbonyl can interact with this proton from histidine, and it is going to form the enol. Now, this in the carbon-carbon bond forming step, the same histidine is still there to remove the enol proton again, and another histidine in its protonated form is placed to donate a proton to the oxygen atom of the ketone. So, here is the histidine which is going to donate the proton to the ketone, and here is the histidine which is going to pull out the proton from the enol, and it is going to give you this product here. Now, histidine, which has a pKa(H) of about 7, is so useful in enzymes, and it can, because it can act either as an acid or a base, right. So, even the hydrolysis of the reactive thiol ester is catalyzed by the enzyme, and the original histidine again functions as a proton donor. So, acetyl-CoA has played its parts in all the steps. So, this hydrolysis, as we have looked at right now, is also catalyzed by histidine. The enolization and the hydrolysis in particular are better with the thiol ester. So, therefore, acetyl-CoA forms a key plays a key role in this entire chemistry. So, CoA thiol esters are widely used in nature, and mostly they are acetyl-CoA, but other thiol esters are also used to make enols. The two enol equivalents that we have met so far are quite general. So, one is from lysine enamine, which can be used for an aldehyde, for any aldehyde or ketone, and the other one is a CoA thiol ester, which can be used for any ester. Another class of enol equivalents, which is the enol ester, has just one representative, but it is a very important one, and so we are going to look at it. So, it is called the phosphoenol pyruvate. Pyruvic acid, which we have looked at in quite a bit of detail in this lecture as well as in the previous lectures, is a very important metabolite, okay, and is basically the simplest alpha keto acid, which is basically 2-oxopropanoic acid. So, it has two carbonyl groups which are adjacent to one another and makes it quite reactive. So, the ketone here is lot more electrophilic compared to a normal ketone, and because of this it also enolizes more readily and the acid is much stronger. So, pyruvate is in equilibrium with the amino acid alanine by an aminotransferase reaction catalyzed by pyridoxal. So, we have already looked at how alpha keto acids are going to be in the equilibrium with the corresponding amino acids. So, here is the equilibrium that is shown here. So, alanine is here, and this reaction is catalyzed by pyridoxal. Nature uses enol phosphate of pyruvic acid, which is phosphoenol pyruvate or PEP, as an important reagent. Okay. So, this can happen by formation of the enol followed by phosphorylation, which is mediated by ATP. So, here is the enol of pyruvate, okay, and since this is an enol, we have already looked at phosphorylation reactions which can occur on OH sites, and so this phosphorylation mediated by ATP is going to give me phosphoenol pyruvate. Now, in fact, this reaction does not occur in nature as part of the glycolysis pathway, but it occurs almost entirely in reverse. It is phosphoenol pyruvate is used as a way to make ATP from ADP during the oxidation of energy storing sugars. So, an enol is a better leaving group compared to an ordinary alcohol, especially if it can be protonated at the carbon. So, the reverse reaction is shown below here. So, here the phosphate attacks. So, you can imagine that this is ADP and the phosphate attacks and kicks out an enolate, which is basically, or an enol, which can pick up a proton and give you pyruvate. So, here pyruvate is a very good leaving group because you have an opportunity for the double bond to pick up a proton. So, phosphoenol pyruvate is made by dehydration. Okay. So, the phosphate is already in place when the dehydration occurs, and so this is enzyme which is catalyzed by the enzyme called as enolase. Okay. So, now let us look at these steps. So, here is 2-phosphoglycerate, which is shown here, and if you can eliminate water from here, then you end up with phosphoenol pyruvate. So, the mechanism by which this process occurs is in the following manner. So, a basic residue in the enzyme picks up this proton right next to the carboxylate and produces this enol, and now this undergoes an elimination, which is very much like an E1CB like mechanism. Again, E1CB is basically E1 conjugate base mechanism, which assuming people have looked at previously, and this elimination occurs in the following manner. So, you have this electrons being pushed here, which then kicks out a hydroxide ion to give you phosphoenol pyruvate. So, therefore, phosphorylation of the enol does not occur, but instead this phosphoenol pyruvate is synthesized by a process by which you form an enolate, which then kicks out through an E1CB mechanism a hydroxide ion to produce the phosphoenol pyruvate. So, this must be the case here as the better leaving group phosphate is ignored, okay, and the worst leaving group hydroxide is expelled. Okay. So, the way we understand this is expelled. So, if we were to produce this substrate by another mechanism, then phosphate is actually the better leaving group, and so because you are going through an E1CB mechanism, this elimination occurs in the following manner. Otherwise, what would happen is that this phosphate would be eliminated and you will end up with a different product. So, this would be an unusual way of making an enol in the laboratory, but it can be used usually to make stable enols. So, an example that we can look at is this molecule here, wherein you have, when you treat this with 1% of sodium hydroxide, you have an E1CB type mechanism that occurs. So, first this proton is pulled out by hydroxide ion to generate this enolate, which then rearranges, kicks out OH minus in an E1CB mechanism to produce this product.